Proteins. What do our bodies need them for? What are they used for? And where do we get them from? In this video, we'll start looking at the structure of proteins, how they're made, and what they actually are. Proteins. You need them for your bodies. They're essential to life. Pretty much they're the necessary structural units of all of your organs, of all of your cells. You have to have them. Now, proteins themselves can be considered a complete protein source or an incomplete protein source. When you're eating something that's a complete protein source, that means you have all 9 of the essential amino acids in that food. Fish is a good example. It contains all 9 of the essential amino acids. Overall, your body has about 20% of it being protein. So that means every cell has some proteins in there because of structure. But you start thinking about things like muscles. That's a lot of protein right there. So what are the functions? We know they're made of amino acids. But what are the functions? Functions are muscle contraction. Your muscles are made of proteins. Your hair and nails made of proteins. Your immune system uses proteins. Your digestive system uses proteins. Pretty much every one of your systems is going to use proteins for one function or another. So it's kind of important to understand what they are as well as what they do. So in total, there are 20 different amino acids. Now, of all 20 amino acids, they vary based on a couple different things. But they're also the same in a couple different things. Their similarity is based on their backbone or their basic structure. All 20 amino acids have a central carbon. They all have a nitrogen-containing group. They all have a carboxyl group. The one thing that varies is the side chain, also known as the radical chain of the R group. This R group, or this side chain, can be as simple as a single hydrogen, or it can be as complex as a 6-carbon ring, all going ring upon ring upon ring. So they can be very large and very complex in all these rings, or just one hydrogen. And they run the gambit in between there. Based on those side chains, it'll determine is that amino acid polar or nonpolar. Is the amino acid fall under a basic or an acidic category? So they have some variation to the amino acids, but it's all based on that side chain, that radical group. Now, of those 20 amino acids, I already mentioned 9 are essential. What that means is the 9 essential amino acids you can't make. You have to ingest them to make sure your body has enough. That means the other 11, though, are non-essential. Doesn't mean you don't need them. It just means the other 11 your body can synthesize and make for the components that you eat. Now, with that being said, 9 essential, 11 non-essential, there are what are called conditional amino acids as well. So conditionally essential. These conditionally essential amino acids can happen during infancy, as a child's growing, and sometime in disease states when you're really sick or have certain diseases. The idea is, in those conditions, these conditional essential amino acids, your body can't make enough to keep up with demand. That means that normally you can make enough, but if you're in these categories, you can't always make enough, so you have to make sure you're ingesting and eating them at this point. But what's really interesting is, we've already mentioned that there's 20 amino acids. Those 20 amino acids make up every single protein we have in our bodies. And what's really crazy, those proteins, they've figured out there's over 100,000 different proteins. Oh, only from 20? Sure. Because those 20 amino acids, when they make the polypeptide chains, they can be three-dimensionally folded on each other, making different shapes, different proteins. The composition, basically which amino acid is in what order, can vary quite a bit. The size of those polypeptides or proteins, for that matter, can vary quite a bit also. The smallest protein is going to be insulin. It's only 51 amino acids long. That's a pretty small comparison. When you look at collagen, which is what's in your skin and a lot in the bones, help give it strength, collagen is a little over 1,000 amino acids. With the largest protein coming in at titin. Titin's found in your muscles, helps give elasticity to the muscles. This one comes in at over 25,000 amino acids. So you have some really big proteins and some very small proteins. To kind of give that perspective, think about music. We're looking at music as in the notes. You have A, B, C, D, E, F, and G. You have the sharps and the flats. But in total, there's only 12 musical notes. If you're saying, well, hold on, all those letters and sharp and flat, don't forget that sometimes a sharp for one note and a flat for another are actually the same note. So in total, there's 12 notes. But how do you get all the music that's written? There's different octaves, different instruments playing it, different intonations, different speeds. Every bit of music is based off those 12 basic notes. Our proteins, everything's based off of those 20 amino acids. So how do you make them? How do you create them? Well, you start off building proteins with DNA. DNA, or deoxyribonucleic acid, is pretty much the stored information of how to do something. That DNA will then go through transcription to make RNA, ribonucleic acid. That RNA can then be translated into amino acids. So pretty much you make amino acids based on what the RNA coding was. You made the RNA based on what the DNA coding was. Well, once you get those amino acids, they start being put one to the next, building a polypeptide chain. Those polypeptide chains can then start to be made into proteins by folding. But it's not just one simple action there. The basic polypeptide chains, making one amino acid, peptide bond, second amino acid, peptide bond, third amino acid, peptide bond. You just keep bonding each amino acid with a peptide bond. This is your primary structure. You can then go into the secondary structure by folding those polypeptide chains. The folding is based on the amino acid interactions. They get folded into what are called helixes or sheets. Think of helix as kind of like a spiral. Sheet, if you fold a sheet of paper back and forth. But then from that secondary, you can go into a tertiary structure. The tertiary structure is now taking the side chains into account. And how do they interact? It'll help to create the more three-dimensional structure that folds further, because are the side chains either repelling each other or are they attracted to each other? You can then take that tertiary structure and make it into the quaternary structure. That's when the different three-dimensionally folded polypeptide chains come together and make the fully functional protein. So here's an example. This one is hemoglobin. Hemoglobin is made up of 4 different peptide chains that were folded, secondary, then tertiary, and now being brought together in quaternary to make hemoglobin. Hemoglobin, by the way, is what attracts oxygen on your red blood cells. So besides all of this folding and structure and everything else, proteins actually have some functionality in food as well. Now, the proteins will help determine the structure of the food, help determine the water retention amount of the food, the texture of the food. So for example, water retention. When you're eating a cake, for example, you might want to say it's a moist cake. That's based on the proteins. Are they attracting water or not? Or the texture of different cakes. Some will be denser, some will be more fluffy and airy. That's based on the proteins that are used and how they create that texture. So a lot of different cooking styles and cooking aspects also come back to how do the proteins interact. Now the way this works is cooking will denature proteins. Basically what that means is denaturing is going to take a protein, cause it to unfold, and go back to its string of amino acids. But once you denature, you can't refold it. That's done. It's over. Because the entire ability to fold has been destroyed. But it's not just cooking that can denature. You can have mechanical agitation. Think of really strong mixing, like a blender. You can have acid, like your stomach's hydrochloric acid. Or you can have high salt concentration. Any of those can cause denaturation of the proteins. Now think about an example. You've seen denaturation, I'm betting. If you've ever cooked an egg, when you first crack it, you have the yellow and the roughly clear around. But as you cook, that clear turns white and solid, and the yellow will turn different shades of yellow depending on the egg. That's denaturing of the protein in that egg. You're denaturing the system. So proteins overall, they've got a lot of functionality in the body, but there's a bit of functionality in food and nutrition and cooking as well. Until the next video.